One of the most common questions in genetic genealogy is, how does a DNA company know that I'm Irish, Scottish, Indigenous American, or African? The answer lies in a concept known as reference populations. When a DNA company provides an ethnicity estimate, it is not reading a label hidden inside your DNA that says English, Cherokee, or Italian. DNA does not contain nationalities, modern ethnic labels, or tribal identities. Instead, your DNA is compared to large databases of individuals whose families have historically lived within particular geographic regions. These groups of individuals are called reference populations. Your DNA is analyzed by comparing thousands to hundreds of thousands of genetic markers known as SNPs, or single nucleotide polymorphisms, against these reference groups. The company uses statistical algorithms to determine which modern populations your DNA most closely resembles. Therefore, ethnicity estimates are fundamentally comparisons of genetic similarity, not direct readings of your ancestral identity. Understanding this distinction is one of the most important concepts in genetic genealogy because it explains why ethnicity estimates change over time and why different companies can produce different results. Think of a reference population as a genetic control group. If a DNA company wants to identify what DNA patterns are commonly found among people from Ireland, it first needs a group of individuals who can reliably represent that region. The company may recruit individuals who can demonstrate that all four grandparents, or sometimes several generations of ancestors, were born within a specific geographic area. The DNA of thousands of these individuals is analyzed to identify genetic patterns that appear frequently within that population. For example, a reference panel for Ireland might identify combinations of SNPs that are common among people whose families have lived in Ireland for many generations. When a customer submits a DNA sample, the company asks, does this section of DNA more closely resemble the patterns commonly seen in our Irish reference population, our English reference population, our Scandinavian reference population, or another reference group? The answer is expressed as a percentage of similarity, creating the ethnicity estimate. One of the biggest misconceptions about ethnicity estimates is the assumption that modern countries have genetically distinct populations. For example, someone may ask, why does my DNA say Scottish instead of English? The reality is that the people of England, Scotland, Wales, and Ireland share thousands of years of shared ancestry. Ancient populations moved repeatedly across the British Isles through migrations involving hunter-gatherers, Neolithic farmers, Celtic-speaking peoples, Romans, Anglo-Saxons, Vikings, and later movements of people between neighboring regions. As a result, there are many genetic similarities among these populations. DNA companies attempt to identify subtle statistical differences between these populations, but the boundaries are not absolute. A person with documented Scottish ancestry may receive estimates that include Scottish, Irish, English, or Northwestern European categories depending on the reference populations and algorithms used by the testing company. This does not necessarily mean their family history is incorrect. It reflects the reality that human populations have always been connected. Creating a reliable reference population is an enormous scientific undertaking. Companies must first identify individuals whose ancestry represents a particular region. Many companies prefer individuals whose grandparents or great-grandparents were born in the same area. This reduces the impact of recent migration and helps identify older regional genetic patterns. Researchers then analyze hundreds of thousands of SNPs across the genome, looking for patterns that appear repeatedly among individuals from that region. Advanced statistical techniques, including clustering algorithms and machine learning methods, are used to identify genetic similarities and differences among populations. However, no reference population is perfect. Every person carries a unique genetic combination, and every population has experienced migration, conquest, trade, marriage, and cultural exchange. Reference populations are therefore models of genetic similarity, not perfect representations of every person who ever lived in that region. A common surprise among DNA testers is receiving different ethnicity estimates from different companies. For example, one company may report 35% Irish, 40% Scottish, 25% English, while another may report 60% Northwestern European, 20% Scandinavian, 20% Irish. This does not necessarily mean one company is wrong. Each company has built its own reference populations using different groups of people and different methods of analyzing genetic similarities. One company may have a large database of individuals from a particular region, allowing it to distinguish smaller genetic differences. Another company may not have enough data to separate those populations confidently and may combine them into broader categories. In other words, ethnicity estimates are only as precise as the reference populations behind them. Many people become concerned when their ethnicity results change after an update. They may say, "My Irish ancestry disappeared. Last year I was 15% Scandinavian, and now I'm only 5%. However, your DNA did not change. The interpretation of your DNA changed? Imagine looking at a blurry photograph. As the image becomes sharper, you may realize that what appeared to be one object is actually several different objects. The same process happens with reference populations. As more people from around the world participate in DNA testing, companies gain more information about regional genetic patterns. A category that was once broad, such as Northern Europe, may later be divided into England, Scotland, Denmark, or other regional groups. Similarly, a previously assigned percentage may be redistributed into different populations as the company develops a more accurate model. Updates are a sign that the science is improving. Reference populations become especially complicated when discussing indigenous populations, ancient peoples, or tribal identity. DNA tests can often identify that a person has ancestry connected to broad indigenous regions, such as North America, Central America, or South America. However, determining a specific tribe is usually much more difficult. Tribal identity is not determined solely by genetics. It is based on history, community, culture, kinship, oral tradition, and legal or sovereign definitions established by indigenous nations. Additionally, many indigenous communities experience disease, warfare, forced migration, assimilation policies, and population disruption after European contact. As a result, the genetic landscape of the past is not always perfectly represented by modern reference populations. This is why a DNA test may suggest indigenous ancestry while being unable to identify a specific tribal affiliation. Some populations are easier to identify because they experienced what geneticists call a founder effect. A founder effect occurs when a small group of people establishes a community and remains relatively isolated over many generations. Because descendants inherit DNA from a limited number of ancestors, certain genetic markers become more common within that population. Examples include isolated island populations, some indigenous groups, certain religious communities, and populations that experienced historical bottlenecks. These distinctive genetic patterns can sometimes make a population easier to identify within a reference panel. However, even these groups are not genetically pure or completely isolated. Human populations have always experienced movement and exchange. The field of genetic genealogy is still developing rapidly. 20 years ago, consumer DNA testing was in its infancy. Today, millions of individuals have contributed DNA data that allows researchers to build increasingly sophisticated models of human ancestry. As databases continue to grow, scientists can identify smaller regional differences and improve the accuracy of ethnicity estimates. The addition of ancient DNA is also changing our understanding of human migration. Researchers can now compare modern populations with DNA recovered from archaeological remains thousands of years old, providing insight into ancient migrations and population changes. However, even with future advances, DNA will always represent probabilities and genetic relationships, not a complete record of a person's cultural identity. The most important thing to remember about ethnicity estimates is that they are scientific models built upon comparison. Your DNA is a biological record of inheritance, but interpreting that inheritance depends on the quality and size of the reference populations used to analyze it. A DNA test can be an extraordinary tool for exploring ancestral origins, identifying relatives, and discovering migration patterns. However, it should always be used alongside traditional genealogy, historical records, oral histories, and cultural knowledge. The most successful genetic genealogists understand that DNA is not the entire story, it is one piece of a much larger historical puzzle.